Inside every single one of your cells, there's a master blueprint for life itself. But how does your body actually read that blueprint without ever taking it out of the vault? Well, that's the incredible story of DNA transcription, and today we are going to decode it together.
Really think about that for a second. Your DNA holds the instructions for building, well, everything about you. It is an unbelievably precious document, and it's locked away for safekeeping inside the nucleus of the cell. The problem? The factories that build all the proteins are way out in the cytoplasm. So you've got this classic dilemma. How do you get the instructions to the factory floor if the master blueprint can never, ever leave the main office? And that, right there, is the DNA dilemma. But the cell has this incredibly elegant solution that's happening trillions of times a second inside you right now. It's a process called transcription.
At its core, transcription is really just an act of copying. The original DNA doesn't go anywhere. Instead, a special enzyme comes along and rewrites a specific part of the DNA code into a temporary, disposable message. And this messenger molecule, we call it messenger RNA, or just mRNA for short.
Now, this mRNA messenger is a little different from the DNA blueprints. For one, it uses a different kind of sugar and it's single-stranded, not a double helix. But the really crucial difference is that it swaps out one of the DNA bases, thymine, for a new one called uracil. This U for T substitution is basically a chemical watermark, clearly marking it as the copy, not the precious original.
So how does this all happen? How does this copying actually work? Well, the process is carried out by a real star enzyme, RNA polymerase. Think of it like a molecular scribe, and it performs its job in a perfect little three-act play. So what are these three acts? First up is initiation, where our scribe finds the right place to start. Then comes elongation, the actual writing of the message. And finally, termination, knowing exactly when it's time to stop. Let's break down each act. Okay, Act 1, initiation. The RNA polymerase scans along the DNA until it finds a very specific sequence of letters called a promoter. You can think of this as a big neon sign that says, "Start copying here." As soon as it latches on, it gently unwinds a small section of the DNA double helix, exposing the code that needs to be read. Now for Act 2, elongation. This is the main event. The polymerase moves down one of the DNA strands, reading it one letter at a time. And for every letter it reads, it grabs a matching RNA building block and adds it to the growing messenger chain. It follows a really simple rule. G on the DNA gets a C in the RNA, C gets a G, T gets an A, and here's the key part. An A on the DNA gets that special U in the RNA. And finally, Act 3, termination. The RNA polymerase just keeps chugging along, building that mRNA strand until it hits another special sequence on the DNA, the stop sequence. This is its signal to stop. The polymerase lets go of the brand new mRNA message and detaches from the DNA. And the DNA, it just zips itself right back up, completely unharmed. But hold on. The story isn't quite over yet. The message that was just made is what we call pre-mRNA. It's basically a rough draft. And just like any good piece of writing, it needs to go through a serious editing process before it's ready to be published, or in this case, sent out of the nucleus. This editing involves a few key steps. First, a protective cap is added to the front, which acts like an address label so the protein factories recognize it. Next, a long tail of A bases is tacked onto the end. This helps keep the message from falling apart too quickly. But the most amazing part is the final step, splicing. So get this, that initial genetic message is actually pretty messy. It's got the parts that actually code for stuff called exons, but they're broken up by these long stretches of non-coding sequences called introns. The useful information is constantly being interrupted by what's essentially genetic noise that has to be filtered out. So the cell brings in this incredible molecular machine to precisely snip out all of those introns and then stitch the exons together into one clean, continuous message. And what's really fascinating is that sometimes the cell can splice the message in different ways, which allows a single gene to create instructions for lots of different proteins. It's an unbelievably efficient system. This whole process, from finding the start line all the way to splicing, has to be incredibly precise. Absolutely perfect. Because a mistake of just one single letter can have profound, life-altering consequences. Let's take phenylketonuria, or PKU, as an example. This genetic condition is often caused by a single letter being swapped in the DNA. A G becomes an A. And that one tiny error is enough to completely confuse the splicing machinery we just talked about. The cell can't edit the message correctly, which results in a messed up, unstable mRNA, and ultimately a faulty enzyme.
And without that one specific enzyme, an amino acid called phenylalanine builds up in the body to toxic levels. This leads to a whole range of severe health problems. We're talking intellectual disability, seizures, developmental delays. It's just a powerful reminder of how critical a single letter in our genetic code can be. But here's the good news. Because we understand the root of the problem on a molecular level, we've actually developed really effective treatments. For people with PKU, a lifelong, carefully managed diet that's low in phenylalanine can prevent the worst symptoms, allowing them to lead healthy, normal lives. So to pull this all together, transcription is the vital messenger service of the cell. It's the complex, beautiful process that safely carries instructions from the protected DNA blueprint out to the machinery that actually builds our bodies. Without it, the code of life would just stay locked away, unread, and totally useless. And that brings us to a final thought. We've seen how a single, tiny letter change can completely alter the course of a life. So as we get better and better at reading and understanding this incredible process, it just opens up a whole new world of possibilities. The blueprint is open, and we are only just beginning to learn its language.